Hello, this is Mr. Philippek, and this video is the final one in this DNA sequence, and we're going to cover both transcription and translation. And first up, we're going to talk a little bit about transcription. And basically, in short order, we're going to go from DNA all the way and convert it to something called messenger RNA, which we talked about in the last video series. And the reason, again, for that is that DNA is too big to get out of the nucleus. And remember that all proteins are made in the cytoplasm. So transcription allows us to convert the recipe here in DNA and eventually, so the recipe here in DNA, eventually to get it all the way out to the cytoplasm here for messenger RNA. And as the slide here says, the whole purpose is basically to create this single-stranded RNA molecule. And again, if we look down here, at this example here, we notice that DNA with its double helix, again, can't get out of the nuclear pores. And so what we do is we end up creating this messenger RNA, which is going to take that message all the way out to the cytoplasm. And so basically, DNA has the recipe book. And, much like maybe the internet or something along those lines, we seek out those recipes and we use our computer to go get them. But in the case of cells, they're going to take that message and send it. It's almost, I think, a better example would be sending a text message. We're taking a message from our brain and putting it on a device and then sending that message out. Now, in order to do this, messenger RNA is transcribed into a different sequence here. And remember, the base pairing relationships for RNA is A now pairs with U, with his uracil, C still pairs with G, and then T has kind of like a one-way relationship to A. We just have to make sure that all the letters are accounted for. And so we call, these sequences where we break them up kind of into triplets here, we call them codons, all right? And then these codons are going to be used in another process called translation, which we'll talk about here in a second, to eventually identify which amino acid that codon will be. And so in this next slide here, we have two different styles of what I like to call battleship charts. We'll call this one chart number 1, and this one over here chart number 2. And basically how it works is, chart number 1, we start in the middle. So here's where we take the first letter in a codon. And then we have the second letters in this next ring. And then the final ring here is this most outer ring here. And we kind of read out. And eventually we end up at an amino acid sequence. So if we take a look at this messenger RNA sequence right here, the first three letters are AUG. And so I'm just going to call this the amino acid sequence here. AUG. So this is the first letter in the codon, second letter, and third letter. And so let's use the circle chart here. And I'll just kind of erase what we kind of wrote on here so we can use the chart again. And so the first letter is A. So here's where A is. And then we're going to go on to U, which is right here. And then the third one is G, which is right here. And we notice here that we get the amino acid methionine, which I'll just abbreviate MET. All right. This also just happens to be the first amino acid in every single protein, because AUG is what we call a start codon. All right. It basically starts what goes on here. We're going to use chart number 2 here to show you how that works. And here we have the code UAU. If you look here on the left-hand side, it says this is where the first base is. So this is where the first reading is going to be. So we want to find the letter U, which is right here. And then the second letter is A, which we're going to read across the top here. And here we have A. And then so we want to read across with U and read down with A and look for our third letter, which is U. And that gives us basically this one here, UAU, which then I so nicely covered up here. The amino acid here we're looking for is abbreviated TYR, which is tyrosine. The last one the last codon here, and again, remember these are triplets called codons here, spells the word gag. And I want to let you use whichever chart you'd like. Okay, and again, I'll kind of erase here in chart 1 so you can see it. Whatever chart you'd like, so I encourage you to pause the video, or just in your mind here, try to figure out what amino acid is going to come up here, and I'll give you a second to figure it out. All right, so again here, I'll use the circle chart here. Here we have G, and then the second letter is A, and then the third letter is G. And so that amino acid here is glutamic acid, so I'll just write GLU. And so that's basically, you know, kind of looking ahead. What we call it going from the messenger RNA to amino acid, that's actually translation. So we've actually taken it a step farther. So transcription is all, how do we come up with this messenger RNA sequence? Okay? And so now, let's talk about translation. So remember, when we go from DNA to messenger RNA, we call that transcription. Translation is going to involve this process of going from messenger RNA to an amino acid sequence, which we just kind of illustrated. And so, again, here we have the codon. And again, those are those 3 base triplets. So if we take messenger RNA and kind of make it in triplets, each one of these right here is a codon. And then each of these codons are going to code for a particular amino acid. And remember, all of this occurs at the ribosome level in the cytoplasm. Okay? And so this idea of going from messenger RNA to translation happens in the cytoplasm. And so what's interesting here is we have 20 different amino acids that code for our proteins. And what's interesting here is that there are 8 essential amino acids. And why are they essential? Well, it's because we have to get them through our diet. Our body cannot manufacture them. So a lot of times when we buy cereal and things like that, we notice that our cereals are fortified with the 8 essential amino acids and all the vitamins and minerals. And so who brings these amino acids to messenger RNA? Well, again, it's transfer RNA. And basically what happens here is that transfer RNA has a series of codons which complement that of messenger RNA, and we call this the anticodon. So it's like the opposite codon. So here, let's just use this example right here. If the anticodon is AAG, what would be the corresponding messenger RNA sequence that would go with AAG? Well, again, remember our base pairing relationships are A pairs with U, C pairs with G, and then T in a one-way relationship will always pair with A. And so if we're going from what would complement A here, it looks like it would be U. So it would be U, U, C. And then we would take this codon and apply it to one of those charts that we showed you earlier to come up with the corresponding amino acid. And so what happens is when we have that match between the anticodon and the codon, we get this temporary bond. And then what happens there is that it makes sure that the amino acids are placed in the proper sequence. So like if we wanted to spell the amino acid earth, you know, we've used this example throughout this video series. This temporary bond makes sure that we basically would have the E in the right spot, then the A, then the R, then the T, then the H, all within the proper sequence. And so what happens is that one tRNA sits there, and what we have here is the growing amino acid sequence. And so the next one comes in here, and I think it's better if we kind of expand this document here. And so again, here is the expanding amino acid sequence, which is basically going to become our protein. And so transfer RNA comes in here. And then we have to use a little bit of ATP. So ATP is brought in here, which provides this bonding of, this is a diamond or a square. And then you can see right here that this energy is used to make this temporary bond, or this bond for this amino acid. And remember, we call those peptide bonds. And then we need a little bit of energy. Again, ATP comes in again to kind of jettison this next transfer RNA and then kind of slide everything over. And we're going to keep doing this and doing this until we reach what we call a stop codon. And this basically does exactly what it sounds like. It tells this ribosomal RNA to stop this process, and it's diagrammed really nicely here. So once we hit the stop codon, which is represented by this little yellow doodad for a better term, we cut the amino acid sequence off, the ribosomal complex disassembles, and then the protein here folds itself into its proper shape. And so here we have, the whole kit and caboodle all put together here. Again, this is the growing amino acid sequence. We call it a polypeptide. Here is a ribosomal complex along with messenger RNA. And again, we're going to keep reading along. And again, here's the anticodon that complements the codon here in messenger RNA. We're going to keep adding on there until we reach that stop codon. And after that point in time, the polypeptide has been formed, and then we have our protein. So I thought we'd end this video with a little bit of practice. And again, I have both charts here so you can practice with both of them. And remember the process from DNA to messenger RNA, we're going to call that transcription. So let's go ahead and transcribe this sequence of DNA and put the proper letters in here, and then we'll take that and translate it into amino acid sequence. So remember, the base pairing relationship with RNA, again, is A pairs with U, C with G, and if we ever run into a T with messenger RNA, that'll pair up with an A. So let's talk about this here. T is going to pair up with, in messenger RNA, that's right, it's A. And then A is paired with U, and then C pairs with G. And then we have C, G, G, C, A, U. And again, to kind of help us organize some stuff, I think it helps if we kind of put a little line between these triplets. And again, these triplets are called codons. So again, we're going to use, either chart is fine, to figure out what AUG codes for. So I'm going to use the chart here on the left and kind of look here. Here we have A, we have U, and we have G. And again, that is our start codon, which is methionine. We'll make our peptide bond here. Next one we're going to go CCG, and I'll use the box on the right here. So here's the first base, which is C. Then we have G, which is right here. And then so we have CGG, and it looks like it all codes for arginine. Very nice. And then the last one is CAU, and I'll let you use whichever box you'd like. So CAU, I'll use the one on the left again. Here we have C, we have A, and we have U, and that's histidine. All right. So again, we've gone from DNA to messenger RNA, which is transcribing, and then we've gone from messenger RNA to amino acid, which is translation. Well, I hope it gives you a nice little review and some practice, and thank you for listening.